Microplastics have been shown to have adverse health effects, so it's important to investigate them within fish that humans eat. Hello, I'm Liam. I'm from England. My project is researching microplastic content within 2 different fish species here in the East Asian. We're trying to investigate if there are see if there's any differences in microplastic levels between 2 commercially important fish species. Maybe they're higher in one than the other. Maybe one particular type of microplastic is more prominent in them. It's just all around broad investigation of microplastic levels within the stomach contents of 2 fish species.
So microplastics are really small pieces of plastic, usually less than 5 millimeters in size, and they come from the breakdown of larger plastics like a plastic toy or a water bottle or a bucket. And in the environment there's lots of physical and chemical interactions that can break it down into super tiny pieces that we can't see, and then it makes it bioavailable to organisms like fish. And it's important to study them because they're found everywhere we've looked for them. So we found them in seawater and freshwater. We found them in sediment. We found them in plants. We found them in fish and animals and food that we eat, like honey and beer. And it's very concerning because we don't know what that means for us and what it means for the environment. So we want to be able to figure that out and find all of the plastic and hopefully clean it up and stop it going into the environment in the first place.
We get fish donated from a fish farm. When they arrive, we must dissect them as soon as possible so that the samples stay in the best condition as possible. From those, we cut open the stomach and the gastrointestinal tract, and we just get the contents of them both in a beaker, and then I undergo a chemical separation and digestion protocol. I use sodium chloride, mixing and sitting it in sodium chloride, to try and separate what's desired, the microplastics, from other stuff such as organic matter, such as fat, to try and dilute it down so that hopefully, ideally, there's just water, well, there's just the chemical and the microplastic. It obviously never gets to that point, but I want to get as close to that as possible. And then I leave the sample overnight in hydrogen peroxide, which corrodes and erodes what remains. And then I do another separation just to make sure. And after a lot of that mixing and stirring and letting it sit in these chemicals, I put each sample through a filter. It gets sucked through via a pump, and then that filter, it's the form of a white disc. I look at that under the microscope, and that's when I visually identify and quantify the microplastic in there, using some software to help me measure it. And then I have data for one sample, and I do that lots of times, and then I have a spreadsheet, I have a data set with which I can perform statistics on. One of the difficulties is that some of the samples, particularly sea bream, are very fatty. They contain a lot of fat. So even after I've done my chemical separation and digestion protocol, there is still fat in the sample. They often take an awfully long time, which slows down the process and slows down how much we can get through. And then when looking at them underneath the microscope, I see a lot of fat which can conceal the microplastics I'm looking at. So that is one of the difficulties. We found that with sea bream, the level of microplastics within the fish goes down as the fish gets bigger. So there's a negative relation between fish size, both in terms of weight and length, and the amount of microplastics inside, which is interesting because you'd think bigger fish eat more, therefore potentially accumulate more. You'd think it would be a positive relation. But it's a negative relation in sea bream, so I'd be interested to do some reading around that to figure out why.